Can you show me? Neck. I just press this one.
Yeah.
Okay.
Yeah.
Good afternoon. I will be talking about, and this was tough, it was painful to make it a 15 minutes or 20 minutes presentation. Thank you for the committee for inviting me. I will be talking about the therapeutic roles of ACE-overexpressing macrophages and their ability to resist the pathological forms of amyloid beta. So obviously, Alzheimer's disease is a very complicated disease, and many different processes are involved. The long-held perception of the amyloid beta as the key pathological manifestation or element of the disease is still holding, although there is a key shift between the different forms of A-beta. So apparently the monomeric A-beta, and specifically the highly amyloidogenic A-beta 1 to 42 alloforms, the longer alloforms, are creating small oligomers, different types of dimers, tetramers, and then seeds and protofibrils into fibrils, and eventually that become a plaque, what we call amyloid plaques, and then neuritic plaques that are involved in, that triggers inflammation and triggers chronic reactive astrocytes and reactive activated microglia that historically, again, perceived as only detrimental, to have detrimental effects on Alzheimer's disease. More recently, it has been shown that the different forms, conformational structures of the A-beta, and specifically A-beta 42, the small oligomeric forms, are showing an increased impact on synaptic integrity. So there is increased loss of synapses, there is altered LTP and impaired cognitive function when animals are introduced with the oligomeric forms of amyloid beta. The genes also show, all of them, currently 30 different genes are involved in Alzheimer's disease. 3 of them are found in the rare familial cases of Alzheimer's disease, and 27 others were all shown to be involved somehow with either higher production or processing or trafficking of amyloid beta. Of course, there's also a tau involved in Alzheimer's disease, I forgot, and of course the chronic inflammation. But many of those genes are involved in immune and innate immune responses, and innate immune cells were shown to be capable in clearing the pathological forms of A-beta. And this is the major focus of this talk. We have previously shown that controlling an immune response achieved by an altered myelin-derived peptides such as glatiramer acetate, which is an FDA-approved drug for treatment of multiple sclerosis, but also MOR45B loaded on dendritic cells, helped attenuate AD-like progression in murine models of the disease in the double transgenic ADTG mice, and regulated neuroinflammation. These studies, our studies, showed that we could, with this immunization, reduce levels of soluble and insoluble A-beta, the plaques. We could resolve scar tissue. We changed the morphology of astrocytes. There is a reduction of neuroinflammation, both the activated and reactive astrocyte, activated microglia and reactive astrocytes. There was an local anti-inflammatory shift. There was enhanced neurogenesis and neuroprotection against glutamate toxicity, as well as there was, and that's the most important outcome, resolution or preservation of cognitive functions. All of these immunizations had a common mechanism by which we were able to enhance the recruitment of monocyte-derived macrophages from the periphery to the CNS that homed into the amyloid lesion sites. Subsequent studies, we either ablated monocytes and then saw that there is exacerbation of A-beta pathology, and other groups had also inhibited the transmigration of monocytes from the blood to the brain and saw that there is an exacerbation or earlier A-beta, amyloid beta pathology. On the other hand, we have shown that when we injected or immunized mice with GA, it boosted the recruitment of monocytes, and here you see it in green. Sorry. GA boosted the infiltrating of monocyte, and it was associated with reduced amount of amyloid beta plaque in the brain. So ablation, the outcome with ablation of monocyte, the outcome was a lot of plaque burden in the brain, and when we increase the infiltration of the monocyte above the normal level, we had reduction of the plaque burden. And these observations led us, and I want to note, that CNS irradiation artificially increases monocyte infiltration. So in the next set of experiments, what we've done is we've done blood enrichment with monocytes that are isolated from the bone marrow, but they are CD115 positive subset of monocytes which are known to be inflammatory monocytes, and they respond to stress signals. Normally in the mouse, there are about 1 million cells that circulate in the blood and respond quickly to inflammation and disease conditions. Here we enriched those population by isolating those cells from young donor mice, and we undergo a process where we enrich the mononuclear cells and the specific subset of monocytes. We inject those monocytes once a month for a period of 2 or 3 months, and we saw first, because these monocytes were GFP labeled, we saw those monocytes transmigrate from the blood to the brain and home to amyloid beta plaque sites. So here in cyan you see the plaque, and here the GFP labeled monocytes, and these are CD45 high or low microglia around the plaque, and the outcome was reduction of amyloid burden, both the soluble and then insoluble. The outcomes were very clear also in terms of cognitive functions following ambulatory and rearing and normal action in an open field. We did Barnes-Mays test, and then we also identified those preservation or retention in cognitive functions following monocyte grafting, enrichment of monocytes, or GA immunization. Those changes were associated with reduction in GFAP astrogliosis, and also we saw that those monocytes expressed in the site around the plaque or surrounding those amyloid lesion sites, they were expressing high levels of MMP9, that is an enzyme that can degrade pathological A-beta. We also have a method to to do serology or detailed analysis of pre- and post-synaptic markers in different regions of the brain. And we were focusing in hippocampus and entorhinal cortex, specific layers, layer 2 and 3. And what we've shown is that those animal models have a significant loss of synapses, both in the hippocampus and the entorhinal cortex. But when we either enriched with monocytes or immunized with GA, we were capable of preserving the synaptic intensity, sometimes back to the level of the wild-type non-transgenic mice. The next step was to ask ourselves whether we see that this effect is due to the increased infiltration of what we call therapeutic or inflammatory monocyte, whether we can enhance the functions of these monocytes by genetically engineering them to overexpress an enzyme that can degrade A beta. And this enzyme was shown previously, angiotensin converting enzyme, or ACE, was shown to be capable to convert the highly toxic A beta 42 alloforms to A beta 40 and shorter, more N-truncated, shorter fragments of A beta that were shown to be less pathological. We combined this approach by first targeting ACE into myelomonocyte. That means that both microglia and monocyte were expressing high ACE, high level of ACE. And what we saw initially is a remarkable prevention of A beta pathology. We thought that we have a genotyping error at first, but we repeated those experiments again and again and again and looked at different time points. And what we've seen is a remarkable reduction. Those are ACE normal wild type AD transgenic mice, and these are mice that overexpress ACE in the monocyte, in the myelomonocytic cells. Again, the reduction was up to 80% at advanced stages of the disease in the animal model. We saw also a reduction in the A beta 40 and 42, the amyloidogenic peptides. Remarkable reduction in astrogliosis, really to the level indistinguishable than the level of the non-transgenic mice. And again, the most important outcome is the cognitive outcome. We see that those animals perform as good as the wild-type animals. These benefits were lost following inhibition in vivo of ACE using ramipril. And we, of course, controlled for blood pressure. Again, I won't, and I don't have the time to get into the details. We did a detailed flow cytometry analysis of the brains and immunohistochemistry, and what we've seen is that there isn't more infiltration, but there is more presence, definitely above twofold presence of these infiltrating monocytes surrounding the plaque. And that the monocytes, and this is we measured by Western blot, these monocytes expressed ACE much more than resident microglia, and those monocytes differentiated into macrophages, and they were capable more effectively to uptake or engulf A beta and clear A beta. And so that's one of the mechanisms that we think that A beta was reduced and the cognitive function was better. And again, it's not only the plaque, it's really the soluble oligomeric forms. The next question was, is it due to ACE-10 microglia or peripheral ACE-10 monocyte-derived macrophages? And the first strategy that we took is to do bone marrow transplantation of ACE-10 marrow. We had shielded this time because we didn't want to have an increased artificial infiltration into the brain due to the irradiation of the brain and causing injury into the brain. So with no changes of blood-brain barrier, with about 40%, when we looked at the brain, about 40% marrow transplantation, we saw about 30 to 47% reduction in the levels of soluble and insoluble amyloid beta, and reduction in GFAP astrogliosis. And since these were GFP-labeled monocyte, we could track them in the brain. And in the brain, we saw that there is increased infiltration of these ACE-10 monocytes. And those ACE-10 monocytes, those specifically ACE-10 monocytes that infiltrated the brain, they had reduced amount of TNF-alpha. So at least in this model, and no changes in microglia, in this model, we saw that this infiltrating monocyte has, at least by TNF-alpha, an anti-inflammatory profile. We also looked at TGF-beta and IL-10. The second strategy that we dissected between or differentiated between the resident CNS microglia and the infiltrating monocyte was to, again, adoptively transfer. And here in this study, we adoptively transfer either wild-type monocytes or ACE-10 monocytes for 2 months, once a month for 2 months. And we saw that both type of monocytes helped preserve cognition. However, there were some aspects of cognition. I'm not showing here everything. There's a lot of data here. We did Y-maze and object recognition. There were some aspects of cognition, and this is the phase, reversal phase. This is acquisition, memory retention, and this is reversal phase, that the ACE-10 macrophages were even better than the regular wild-type macrophages. And then we asked ourselves how those ACE macrophages are different than the wild-type macrophages, and we looked at different pathological forms of A-beta, and we looked at binding, recognition, internalization, degradation, and intracellular, some aspects of intracellular processing of toxic amyloid beta. This was done in vitro, either by using wild type or ACE-10 donor mice. We took the bone marrow, we cultured the cell and derived them in 7-day culture into macrophages and did binding, phagocytosis, degradation, and other assays. Here you see where we use fluorescently labeled A-beta 42, the uptake, the clear uptake phagocytosis by CD68 positive macrophages. We also uniquely, with collaboration with Dave Teplow and Eric Hayden from UCLA, we obtain structurally defined by SDS-PEI gels and by transmission electron microscopy. We use defined forms of oligomeric A-beta, protofibrils, and preformed fibrillar A-beta. And we looked at first their impact on synapses. So definitely we get 50% in 12 hours, 50% loss of synapses in primary cortical neuronal cultures following exposure to fibrillar forms of A-beta, and then 70% loss of synapses in response to oligomeric forms of A-beta. We see that the ACE-10 macrophages obviously can uptake A-beta better after 5 minutes, 30 minutes, 15 minutes. We've done it by immunohistochemistry and flow cytometry, and following lisinopril that, again, inhibits the catalytic domain of ACE, we lost this benefit of increased uptake of A-beta by macrophages. We looked at binding, so either it's technically hard to check for binding of oligomeric A-beta, but clearly the ACE-10 macrophages can bind fibrillar and protofibrillar forms of A-beta. All the ACE-10 macrophages are capable to uptake after 15 minutes much more A-beta 42 in all their different forms. So both oligomeric, short fibrils, and fibrils. We looked at a panel of scavenger receptors. These are a pattern recognition that are expressed in the surface of macrophages, and I'm just showing you some of them. And SCARA is a class A scavenger receptor. It has been associated with facilitation of A-beta uptake with regards to Alzheimer's disease. We saw that SCARA affected only fibrillar, but not oligomeric or protofibril, but mostly fibrillar forms. SCARA is innately, before exposure to A-beta, is highly expressed on these ACE-10 macrophages, but also after fibrillar A-beta. And here we looked at other forms, oligomeric. There was no difference between SCARA expression, and there is a clear correlation between a SCARA surface expression and surface binding of A-beta. So as much as you have higher expression of SCARA, you have higher, it predicts better ability to bind to fibrillar forms of A-beta. We looked also at 36. CD36 is another class B scavenger receptor, and it has been implicated in inducing intracellular signaling and processing of amyloid beta. And we're definitely seeing differences, remarkable differences between the ACE-10 without A-beta and with the 3 different forms of A-beta, also correlating and predicting better uptake and processing of A-beta. We looked also at endosomes, early and late endosome markers. Here you see some data regarding the EEA1, and definitely both two different analyses, either of the number of EEA1 positive vesicles co-labeled with A-beta, and the percentage of A-beta positive in EEA1 early endosomes. We see in all the 3 different forms that ACE-10, ACE-overexpressing macrophages are capable to uptake into endosome the 3 different structures. There's definitely differences between the structures, the chemical structure or conformational structures, where we see the highest level of co-labeling with the fibrillar and the lowest level with the oligomeric forms, soluble oligomeric forms of A-beta. There is an anti-inflammatory profile to the ACE-10, and the previous speaker has talked about the ED2 or CD163. So both arginase 1 and CD163 was remarkably increased in ACE-10 macrophages, and iNOS and nitric oxide and MHC class 2 were reduced. Another parameter that showed us a pro-healing or anti-inflammatory profile is the cell elongation, and we did a detailed analysis on cell elongation that previously was associated with the alternatively activation of macrophages, and we see that ACE-10 macrophages has an alternatively activated form shape, cell shape. Also, the ability to resist. So macrophages that overly express ACE are more resilient to cell death induced by toxic forms of A-beta. And also they are capable, and this is predictable because of the high expression of ACE that can degrade A-beta, they are indeed capable in 18, 20, and 23 hours capable to facilitate degradation of or enzymatic cleavage of A-beta. Lastly, we are now currently looking at co-cultures, primary co-cultures of cortical neurons or hippocampal neurons and primary bone marrow-derived macrophages, and we're looking in details on pre- and post-synaptic markers and their colocalization. And what we see is that there is a clear protection, complete protection for the fibrillar forms by macrophages. There is a somewhat protection, but not complete protection, also from the more toxic form, which are the oligomeric forms of A-beta. And now we're asking ourselves, currently we're looking at ACE-overexpressing macrophages and whether they can exhibit increased protective to neurons and synapses. In summary, we showed that the subset of CD115 or CFIMS positive monocyte can fight the pathology associated with Alzheimer's disease. Moreover, the ACE-overexpressing macrophages, so combining an enzyme that can degrade A-beta with inflammatory monocyte was highly effective in reducing many abnormalities seen in Alzheimer's disease, including regulation of local inflammation, and this was dependent on the catalytic activity of ACE. ACE definitely affected, in our studies, the macrophage phenotype. What we see is a anti-inflammatory. I don't like to call M2 because we now know it's very heterogeneous, but what we've seen is the panel that I've showed you and other markers on monocytes and cytokines that we see that are different. Scavenger receptors are definitely higher in those macrophages, ACE10 macrophages, and they have elongated cell structures that can entail for increased healing processes. In terms of therapeutic aspects, we've shown that either bone marrow transplantation or adoptive transfer of these monocytes into symptomatic transgenic mice reduced cerebral A beta and the different forms of amyloid beta. It reduced the GFAP astrogliosis, and it retained cognition. Our studies also have implications for the use of anti-inflammatory drugs that obviously failed in Alzheimer's disease. And again, that's, I think, because it just generated a general suppression of inflammation, both the good and the bad. So there's definitely a detrimental inflammation locally in the brains of Alzheimer's disease, but we at least and others and more around the world, groups are showing that there are also good inflammatory cells that are critical for resolution of the pathology of Alzheimer's disease. Our studies also have implication for lifelong use of ACE inhibition, and ACE inhibitors are also, some of them infiltrate the brain and some of them work only in the periphery and some of them work in both periphery and the brain. So these are considerations that we should discuss, and we should take a careful examination of them as we look at the risk for Alzheimer's disease. This is my group, and some students are here highlighted staff that are that their data are shown in my presentations. Joseph Koronyo is also collaborating with us, and we have many collaborators. We were also the pioneer group that showed the pathology of Alzheimer's disease in the retina, and the retina actually allows us to follow up migration of monocytes and look at monocyte infiltration in live animals by non-invasive retinal imaging. So that's also, I didn't have time to talk about this, but this is also a major focus of our laboratory. And we are funded by NIH, but also by private foundations and others. Thank you.
Some questions?
Yeah, I have a couple of questions. The first one, I might miss it about your